Swayam Prabha. Digital India. Educated India.
Hello everyone. The topic for our group is Adventures in Synthetic Biology, and this paper basically gives an overview of how DNA works and on the basics of synthetic biology. So it introduces synthetic biology in the terms of bacteria, and it introduces genome sequencing, and also introduces the concept of biobricks and how we can take parts from biobricks and integrate into other bacteria and give it new features and stuff. So in essence, it introduces us to the concept of producing transgenic strains of bacteria. The paper doesn't go into details into how this is exactly done, and that part is done in an abstract way, and this is mostly because of the format the paper is in, that is a comics. Going on, it introduces a problem with creating the transgenic strains by just introducing genes from different bacteria through biobricks. You can see that in the comics, which is basically we have no regulations for whatever is happening in the bacteria. The process keeps on happening, and we need a feedback loop or a regulation to make it stop eventually. On that topic, it introduces inverters. So what is an inverter? An inverter is essentially a NOT gate, that is a biological NOT gate that is made up of 4 components, that are ribosome, binding site, repressor, terminator, and operon. Drawing parallels to programming, the inverter basically need to tell the genomic machinery when to stop the production of a particular gene. And we are adding feedback loops to control the output of the system. Okay, in this slide, you can see the parts of the inverter, the parts of the biological inverters. And the repressor is gene that codes for the particular protein which can bind to the operator and modulate the expression of the gene by downregulating when it is bound to it. That has the ability to downregulate the flow of the RNA polymerase along the DNA strand, potentially to the point where no transcription takes place and the entire process just stops. This introduces another problem. When we are regulating one part of the gene, we cannot basically make it interact or interplay with the other parts of the gene. So in this, the paper introducing this as another concept of POPs, which is a common signal, which is kind of common signal for a carrier in genetic devices. POPs is basically polymerase, is a measurement of RNA polymerase per second. The paper tries to make an interface between different systems by linking this repressor protein repressor protein POP signal to as the input for the next system. The main highlight of this paper is that it promotes gamification of the whole biotech thing, and it is in an alternative format, that is as a comics. So this gives certain advantages and also certain disadvantages. The advantages include more accessibility, and it can promote interdisciplinary research because it makes it very easy for people from other fields to understand biological concepts. Also, this provides a lot of graphics. So if you are having issues with visualizing the content or how things happen inside the cell, it kind of helps you visualize things. Okay, consequently, it also involves a bit of disadvantages, but most of the disadvantages arise because we are not the targeted audience of this paper. The targeted audience of the paper is someone who is new to biology. So for biologists, this paper doesn't really provide a lot of information that we may want or need, and we have to look at other places. And consequently, it raises a lot of questions like how can we actually measure POPs? POPs is given as a concept and it is left as an open question in the paper. And we basically get no way of actually taking the input signal from there, output signal from there, and using it as an input signal in something else. That's basically it. Thank you.
Your critical appraisal of this.
This introduces a very interesting concept that can link computer science and biology in a very simple manner. Like I am from a CS background, so it really helps me understand the thing because you have a common output signal that you can normalize and you link with basically everything else. And you can use the output signal as an input signal in some other system. And in another way, it actually, if you're looking at it from the perspective of a molecular map, it actually simplifies the whole thing because you don't have to look at the concentration of the end product because the concentration of the end product is dependent on a lot of factors. You have other pathways that are creating the end product. You have degradation in the end product, and if the organism is living in a community, you have other different types of organisms potentially producing the same end product. So in this case, if you can actually quantify POPs and capture the signal, you can create transgenic strains without understanding the entire metabolic map. And in some cases, understanding the entire metabolic map is a very resource intensive process that is best avoided, I guess.
Okay, very good. Any other question or comment for your colleague? No? Okay, well, thank you very much.
Sir, when we discuss the paper, we do have a question for you. Is there actually any way to capture the POPs from a cell, like actually get the signal?
Very good question. Very good question. So POPs so far is a kind of concept. Measuring it is very complicated. And well, in fact, there are a couple of methods. I may discuss them in one of the talks, but it's very complicated. So there's no easy way to see how many times the polymerase goes. Because what happens also is that at the level of single cells, the transcription initiation is an stochastic process. So it's not that, every time that you have a polymerase, it goes and it goes smoothly. No, it goes like, it goes in and out, and only with a probability, then it goes in one direction. And then even sometimes it progresses in one direction and goes back. So it's at the molecular level, transcription is a very stochastic process. So, and therefore counting the number of times that the polymerase goes through a promoter as an absolute measurement is not easy. Okay, and there are basically a couple of methods. None of them is direct. Both of them are indirect. But conceptually, POPs is a parameter for sure that should exist, even though we don't know very well how to measure it. Okay, but very good question. Excellent.
Thank you, sir.
Okay, thank you very much, and maybe we can proceed with the next presentation.
I'll be presenting the work done in this paper called Total Synthesis of E. coli with a Recoded Genome. So we know that in nature, a genome has 64 codons which encode 20 amino acids, which means there is some redundancy. What the authors in this paper have done is they have replaced two sense codons by their synonym and a stop codon with its synonym, so that the resulting genome has only 61 genome, where 59 codons code for 20 amino acids, and the rest of the two are stop codons. And they have done this in a strain of E. coli. And this is the first genome-wide recoding that has been reported. So they have replaced the serine codons TCG and TCA with their synonyms and the stop codon TAG to TAA. And in replacing these codons, they have taken care of overlaps by inserting some synthetic sequences which totally decouple the overlaps, and then they go about replacing with the synonyms. And after this, they have separated the genome into 8 sections, each having 0.5 MB approximately. So the segments that they separate from the genome, they are further segmented into 100 kb fragments, and these fragments they put together using bacterial artificial chromosomes, and they assemble it inside yeast, and they use a method called REXer, which is replicon acquisition for enhanced genome engineering through program recombination. Here, this method is used for replacing synthetic genome into back. And then, it is CRISPR-Cas9 and lambda-mediated recombination. And using this REXer, they have generated 5 strains or 7 strains having only one section replaced by the synthetic DNA. And then they have used this method called genesis, which is genome stepwise interchange synthesis, where they assemble all the strains together so that, as you can see in the figure, each section is clockwise replaced in subsequent steps from the recipient, the donor to the recipient. And then finally, after replacing each section, finally the synthetic genome A to H, all the segments have been replaced by PIC, which is the synthetic part of it. And the resulting genome is, they have named as SYN61, since it has 61 codons. Now, they have compared the 2 genomes, the original MDS S42 strain of E. coli with the SYN61. What they observe is the new genome has 65% more AGT and AGC codons. It grows 0.6 times slower than wild type, and then the cells have been reported to be slightly longer, and the proteins seem to be similar. One interesting property that the new genome has is it is no more toxic to some non-canonical amino acid CYPK, because it is genome that is recognized in the new genome. So the tRNAs which are responsible for translating this is required, which was originally essential in the original E. coli. And so some particular tRNAs which translate CYPK. So the key conclusions of this paper are: this is the first time that they have replaced an entire genome with synthetic codons. And as you can see in the figures, this is the largest genome replacement done so far. And then they have also the maximum number of codons in a single organism, and the number of mutations that are non-programmed mutations, that is the errors that occur given the number of target codons they are changing, it is very low, which means it is very it has very low error. That's it.
So again, the question, some critical appraisal.
Okay. So I do not understand. What if the mutation or this change could make the strain very dangerous or something like that? Is there any such possibility, or is it something we know very well would never happen?
Well, I think that this article has like various, consequences. First of all, is to show muscle, muscle. I mean, to say, well, we are able to do it, even if the follow-up applications are not crystal clear. I say, well, we are able to take the entire genome of E. coli, the whole genome, and resynthesize it completely through chemical means. And this is conceptually that's very remarkable, because basically we are using the hardware of E. coli to run our software. So we have synthesized. Well, they have synthesized the DNA molecule, but I mean in various steps and so. But at the end, it's all synthetic DNA, and in my opinion, that's very remarkable. So that's number one. That's to show muscle, and that's very important. And number two, by releasing or by liberating or by as some people say, emancipating some codons, then you can reassign these codons for making proteins with non-natural amino acids. I think that in one case, there was a codon that was reassigned for incorporation of a non-canonical amino acid, if I remember one of your slides. So by introducing one more amino acid, then obviously you can multiply the number of proteins that you can synthesize with new functions and perhaps to discover new functions. So that's, but I would say that this paper is more a conceptual paper than a contribution for practical applications. The practical applications are not immediately visible, but the fact that you can, in this case, do show that you can do it, that you can take a complete genome and entirely do it. There's a counterpart these days on with the chromosome of yeast. So yeast is also now, every chromosome is being synthesized, and soon they will have a yeast cell that is entirely synthetic from the point of view of the DNA. More questions or comments? No? Well, thank you. Very good job. No.
So, this is a paper on environmental signal integration by a modular AND gate. So, microorganism uses this genetic circuit to integrate the environmental signals, and they need sensor to get the inputs for that. Like it's the same as we there is a speaker, so we can, or ears, we can hear that there is a speaker, and we can get the direction because of ear, whatever we are listening to, and or because of eye we can get this is the particular thing that's the origin of this thing. So they require sensors for receiving information and circuit to process the inputs. Bacteria uses there are a variety of mechanisms to sense their environment. So some of them uses a two-component system and some of them uses one-component system. But a two-component system is better. It's like having multiple sensors, like same as we have ear, nose, and vision. So it's like this. So it's a way we'll get more specific information about our environment. So after this building these circuits, we'll be able to encounter unnatural environments. Like we'll get more information because we have two inputs or multiple inputs. Then we'll be able to get more details about unnatural environment, and we'll be able to integrate those signals. So till now, different logic gates have been built using biological components, but those were not modular, and for that they use transcription factors and regulatory elements or protein-protein interactions. But because those were not modular, so it was very difficult to change the input, because we'll change the input, the whole reaction or whole circuit will change, and we won't get any output. So it was the first one that if we change our input, then we'll get a different output. So it was the modularity facilitated the incorporation of circuit into different genetic programs. So for this AND gate, inputs and outputs were choose to be promoters, because in a cell, if the inputs and outputs are promoter, then we can either use it for output, we can use it for to express a gene, or we can use it as input for other circuit. So for this one, they use T7 RNA polymerase gene and one suppressor tRNA support. And arabinose and salicylate used as input connection. For it was the medium for they used for, and GFP was our main aim for our green fluorescent protein. That translation of this protein was our main aim. So for demonstrate this behavior, that if we change our input, they will get the output. What they did, they change input from T7 RNA polymerase and subject to current signal A1 and magnesium limitation. So after that, what the output was, it is switched on invasion gene that was killing mammalian cells. So it was the demonstration that we got the modular circuit. And in transcription based system, like it's convenient to be promoter, because we can use it for a next circuit, or this express another gene. So it was very crucial that using a promoter as input and output. So this is the processes we are using, and in what's happening in wild type, when this T7 gene was going, it has this stop codon TAG. So it was it was released by factor 1 and resulting in translation termination. But after adding sub-D, what happened? Like it was translating this this TAG to serine and translation resumed. So it was like if the both inputs are not available, it won't work because translation will stop. Then for circuit dynamics, they used this subgene in under a controlled salicylic activated promoter, and T7 gene was placed under arabinose inducible promoter. So in that, when they demonstrated this one, what they got that strong ribosome binding site, and it was not functioning as AND yet. It was because of this arabinose, because it was not dependent on this sub-D. It was not dependent on salicylic activated promoter. It was dependent on this one. So it was a very the circuit was always inducible by arabinose. So they for that they use some introduce some mutation in that so they can handle the situation and make it a perfect AND gate. So transfer function, they derived a model they derived a mathematical equation for that, because transfer function model, with the help of we can discuss this equilibrium state and steady state of this circuit. So it was the equation G by Gmax and I1. These, I think they didn't clearly mention what are these I1 and I2. I think these are the concentration of inputs, and G by Gmax, it was the Gmax was the maximum fluorescent we got, the intensity of maximum fluorescent we got, and A and B they used the different method. They use one component, double component to calculate these values. And I think for A they got 50 plus minus 30, and for B they got 1000. And it didn't clearly mention like what are those parameters for and how they are affecting those. So, and to calculate, they used I for one dimensional for I1, I2, and for fluorescence measure function of inducer concentration. With the help of this, they demonstrated, and for this two-dimensional data they used to calculate G by Gmax value. And with the help of the data they got, they plotted these two graphs. Here, this first this one here it is showing that when we are using and this salicylate and arabinose concentration. So for circuit modularity, that's why the inputs from two natural, this quorum sensing and Mg+2 responsive inputs. So the output was the output transferred from GFP protein to invasion gene that was killing mammalian cell. So it was the first ultimate goal. It was the first, I think, first and modular logic gate. That was like if we switch inputs, we'll getting the different outputs. So and it was the first, unlike electronic circuit, where the spiral wiring of a circuit determines the flow of information through the circuit. Intercellular circuit prevent cross communication through specificity of biochemical interaction. I think that's it. Thank you.
Thank you very much. It's very interesting. So, any typical appraisal?
Yeah, it was very interesting. Like we are using the same circuit, but using different inputs, we are getting, like in a biological system, we are getting different outputs, because we use a particle sets of reaction and all, and we are somehow able to change those, and we are getting the different. We are using the same reactions, we are getting a different output. So it was very interesting, and like I'm interested in how in the future we'll use these circuits in this like instead of electronic signal circuits we'll use for computing other things. So it's very interesting.
Any final comment? Okay, well, we're running later than the day, but it's time for thinking. Okay, ladies and gentlemen, we're having this day today, and for that tomorrow we will follow up with the spirit. Okay, see you tomorrow, and please ensure that you read the paper. Okay?